Hello everyone, my name is Arzoo Patel and today I'm going to be talking about the extraction of amyloid beta and its epimers and analysis through liquid chromatography mass spectrometry. I've been working on this project in Dr. Armstrong's lab along with a few other lab members. As we know, amino acids are the building blocks of proteins, antibodies, peptides, and various other protein structures. Historically, only L configurations were thought to be involved with mammalian physiology, but research indicates that D amino acids, such as D aspartate in proteins, induce aggregation and loss of functions, leading to age related diseases such as cataracts and Alzheimer's disease. As we know, Alzheimer's disease is characterized by deposition and aggregation of amyloid beta, which is used as prognostic biological marker for Alzheimer's disease diagnosis. It has been suggested that post-translational modifications of these amyloid beta, including isomerization and racemization, among others, enhances amyloid beta aggregation. An example of an isomerization of an amino acid residue would be L to D, L aspartic acid to D isoaspartic acid, and racemization would be L aspartic acid to D aspartic acid. While epimeric peptides would differ only at one chiral center from each other. Previous research indicates that senile plaques of amyloid beta aggregates from Alzheimer's disease patients contain 20% isomerization of L to D isoaspartic acid at aspartic 1 residue, while aspartic 7 residue sees a 75% change. In this study, we will be looking to extract 18 different epimeric amyloid beta peptides, and we will be developing the method on standard peptides, which will eventually be transferred to plaques from Alzheimer's disease brain. To extract the epimeric peptides without bias, we will be using immunoprecipitation, which uses antibodies to extract antigens, in our case the amyloid beta peptides, from a matrix. To determine which antibodies will efficiently pull out all of the 18 epimers, we will be testing out 5 different antibodies with different epitopic binding regions. Epitopic binding region is the region on the antigen, which in our case is our amyloid beta, that the antibody binds to. Here I have highlighted the epitopic binding regions of the antibodies A through E. I have also mentioned the location of aspartic acid residues at 1, 7, and 23, and serine residues at 8 and 26. For analysis of the peptides, we will be using a selective and comprehensive method that can easily detect both aberrant and normal amyloid beta peptides simultaneously using high-performance liquid chromatography mass spectrometry, which has been previously developed in Dr. Armstrong's lab. This method uses hydrophobicity as the primary contributor to retention. We have modified this existing method for our purposes. We'll be using BioShell IgG 1000 angstrom C4 column with step gradient mobile phase containing acetonitrile and water. The in-depth method is listed here. We're currently working with standard peptides. To prepare them, we will be diluting the peptides to 25 microgram per milliliter using 10 millimeter boric buffer. Here I'm showing the immunoprecipitation process that the samples will be going through. You would start out with your antigen, your amyloid beta, surrounded by a matrix, in which you would add your antibody. Next, agarose beads would be added. These agarose beads have a protein AG attached to it. This protein specifically attaches to antibodies. This will form your antibody, antigen, and agarose bead complex, in which after you would conduct a wash cycle, which would remove the remaining matrix. And then the last step would be elution, which would release your pure antibody and antigen. In this figure, I'd like to show you the preliminary result of the 18 peptides and the 5 antibodies. Peak areas have been graphed against the 1 through 42 amyloid beta standard and the 18 different epimeric peptides. The 5 different colors indicate the 5 antibodies that were used. The results show that there is a variation of peak area for each peptide and antibody. There is a notable increase in peak area at peak 11, which is actually likely due to the use of a new chromatographic column. This large increase in peak area shows us that there is likely a systematic error that is resulting in irreproducible results. There's also a subsequent decline in peak area, likely due to protein buildup in the column. Overall, the preliminary results show a possibility for project success, but improvements in method need to be taken into account to remove the introduced systematic errors. It also shows us that we need to take into account what antibodies we used when conducting immunoprecipitation for sample preparation.